Hello everyone, this is Group 18. We are going to talk about the C3convertase. The flow of the video is as follows. Definition. First, we will explain about the complement system. The complement system is a system that causes non-specific humoral immunity and is one of the most important systems in innate immunity. Proteins C1 to C9 play a major role in the complement system. These proteins are produced in the liver and circulate in blood vessels. When these proteins are activated in the complement system, they cleave and activate the next protein, which in turn cleaves and activates the next protein, and so on in a series of reactions. This is how the pathway of activation in the complement system works. There are three pathways of activation in the complement system, and the first activation of C1, C3, or C4 precedes the reaction and can cause an immune response. C3convertase works by cleaving and activating C3 in the reaction of the complement system. Cleaved C3 becomes C3a and C3b, which are activated, and this allows the reaction of the complement system to proceed. C3a and C3b also have their own functions. Biological effect. Next, we will discuss the function of C3a and C3b. C3a, also known as anaphylatoxin, plays an important role in the immune response of the complement system, acting on mast cells to facilitate the migration of phagocytes, such as neutrophils and macrophages, to the site of inflammation, thereby promoting innate immunity. However, too much C3a can cause anaphylactic reactions. C3b binds to the bacterial cell surface. Phagocytic cells such as neutrophils and macrophages can then easily recognize C3b bind bacteria, and phagocytosis can easily occur. This effect of facilitating phagocyte recognition and phagocytosis is called opsonization. Two types of C3convertase. As mentioned earlier, there are three pathways in the complement system depending on the protein that is activated first. In fact, there are two types of C3convertases depending on the pathway of the complement system. The classical pathway in which C1 is activated first, the alternative pathway in which C3 is activated first, and the lectin pathway in which C4 is activated first. The classical pathway is described below. The classical pathway is initiated by the binding of C1 to IgG antibodies bound to bacteria. C1 activates C2 and C4, which act as C3convertases when C4b and 2a form a complex. In the lectin pathway, mannose-binding lectin produced in the liver binds to bacteria and converts them to C4. The alternative pathway is initiated by the binding of C3b produced by the other two pathways or by a mechanism called spontaneous hydrolysis to the bacterial cell surface. It also involves substances called factor B and factor D. Bb, which is produced when factor B is cleaved by factor D, forms a complex with C3b. Finally, there is a supplement. In addition to C3a and C3b, there are other functions of the complement system that are involved in immune responses, namely C5a and C5b, 6, 7, 8, 9. C5a, like C3a, contributes to the migration of phagocytes to inflammatory sites. C5b, 6, 7, 8, 9, called the membrane invasion complex, disrupts bacterial cell membranes by puncturing them. This reaction is as effective as neutrophil phagocytosis in defense against bacteria. This concludes our presentation.